Okay, thank you. How do I turn this on? There's a little thing right there. You can just flip the switch and move it. There you go. Am I blasting? Everybody good? Okay. We'll see. I may go overboard if it starts screeching. I'll try to be quieter. So today I'm Jamie Sherman. I'm the barley breeder at Montana State University. And I want to talk to you about some preliminary data about. I'm just trying to get that bar off the top for you. And have you hands? I've noticed people are having trouble. Yeah, you should be able to use the arrows. This? Yes. Okay, we don't. I'm not sure why I can't get that off. It's okay. I want to talk about some preliminary data we have on the microbiome project that we're working on. Should I take this off? Maybe set it right here and just see. We'll let you do it. We'll let her fiddle with it. Oops, made it worse. This is going red. I wonder if there's a battery issue. Try now. How is it? Maybe it's just certain genetic types that make it reverberate. Try now. How is it? It's not on, I don't think. That's all right. You can hear me.
Yeah,
I say
Jamie and I are loud. Say talk again.
Hello?
Is that coming through at all? It is.
Is it better? Let's leave it off. If you can't hear me, raise your hand eventually, but hopefully it won't screech. Okay, so I'm going to give you some preliminary data on a GWAS project to associate the genetic ability of barley to attract beneficial microbes. And this project was funded primarily by USDA, an AFRI grant. But I want to appreciate all of our partners, our funding partners, because honestly, everything comes together to make the program happen. So, but the USDA grant was primarily headed by a microbiologist at one of our research centers, Dr. Jed Eberly. But you can see there the list of people working on the project, including myself and Jessica Williams, who's going to actually show you the data in a moment. So I asked Jessica to come and talk for a couple of reasons. First, she's a really good at explaining these sorts of things. It's good experience for her, but she's the one doing most of the work. So, but I just want to set the stage for us. So everybody is wanting to talk about sustainability, or maybe the new word is regenerative ag, resiliency. And so researchers are interested in it, companies are interested in it. And so the way we've been addressing that primarily as breeders is working on genetic improvement. So we've worked on abiotic stress tolerance. We've heard some talks about that. We've been working on improving nutrient use efficiency, and we've been working on genetic resistance to pests. And you've heard talks about all of those things. Now, the microbiologists, what they've been doing is trying to isolate some of these beneficial microbes. So they isolate them, they culture them, and then they form a company and provide them as a product that growers can use as a seed treatment or some other treatment. And that has some research behind it that shows that it has a positive effect on resiliency. The problem with that is not all beneficial microbes are culturable. There's a lot of beneficial microbes that we don't even know about. And so can we use the barley genetics to attract the beneficial microbes that are already in the soil to the barley plant and take advantage of what's there naturally so we don't have to have this product? So that's where we're coming from. So many of you may not be microbiologists. So this is, if you know about this, you can just go to sleep at this point. But all microbes in the community are not necessarily beneficial. So we can have a microbe that is neutral. As we already have talked about, we can have microorganisms that are pathogenic. But what we're really focusing on are those beneficial microbes that somehow promote plant resiliency or plant performance. And so these are some of the ways that they do that. Now, as people have worked on this, they have found that the microbiome communities can vary for a lot of reasons. One important one is environment. So our focus has been on the root microbiome, the rhizosphere. And so you can think about variations in the soil, the type of soil that you have, how you've managed the soil, what was the previous crop, how fertile, how much fertilizer have you put on the crop. All of those different aspects can impact the microbiome. Even something simple like till versus no-till has a huge impact on the microbes that are present in the rhizosphere. But also we see variation in the microbiome based on what crop is present or plant species. Which of the plant organs are involved? So the microbiome of the leaves is different than the microbiome of the rhizosphere. But from my perspective, the important difference that I'm really trying to get at is the plant genotype. So if we're going to use the plant's genetics, or am I going to breed for attracting beneficial microbes, then there has to be a variation in the genetics of what microbes are attracted. Some of the questions that I had about this from the beginning were, is there a difference in attraction between beneficial and pathogenic microbes? Like if we're going to attract microbes, are we going to attract everything? Or can we differentiate? Is the genotypic differences dependent on the microbial community? In other words, so say I have a gene, but maybe it only works if certain microorganisms are present. And so then that would make it a problem as effective across a bunch of different locations. So we have all of these questions, which we're beginning to address with this GWAS project. Just to give you a little overview, we're going to talk about more of this as Jessica presents the results. But these are the benefits that we could get from these organisms. Everybody probably already knows about nitrogen fixation because we think about it with pulses. But there are also nitrogen fixers that are free living. So you don't have to have nodule formation to benefit from nitrogen fixation. So there's some evidence of those genera. In Montana, one of the things that we've observed, not surprisingly, is low temperature loving microbes. Now, these could benefit us because they seem to also provide stress tolerance and promote plant growth, so that we could take advantage of those. There's other growth promoting microbes. We don't really know how they work. They sometimes produce plant hormones to promote plant growth, but they also may have other means of doing that. And then an even more general way to think about it are all the microorganisms that help recycle nutrients. And this could help turn over nutrients and make them available to the plant. One of the surprising things that Jessica will talk about is we have a lot of bacteria predators that we're seeing in our results. And so these are going to focus on these bacteria, eat other bacteria. And so they can help recycle some of these nutrients. At first I was thinking, well, we don't care about those predators unless they were going after the pathogenic bacteria, but in fact, they could help recycle nutrients too. So we have some evidence of those being important in the Montana system. So just to give you a little bit of a big picture look at what we're looking at is if you look at the soil, and we're calling it the non-rhizosphere, or sometimes I might talk about it as the bulk soil. So it's the soil not associated with the plant, not associated with the roots. That soil, the community in that soil is primarily organized by the organism and then the other microbes that are present. What we're focused on primarily, we're comparing the two, but what we're really interested in looking at is the actual community associated with the plant roots or the rhizosphere. And this community is organized by the environment and the microbes present, but has an added organization of the plant and the chemicals that the plant gives out into the environment that then can attract different microorganisms. And that's where we think the genetic potential is coming from, the variation in these exudates that are created by the plant. We haven't gotten that far. We don't have genes that we can talk to you about because this is preliminary data. But what we have right now is genetic locations that we hope can get us to the point of looking at a gene. So what are our challenges in doing this work? Well, our first challenge was, will we even see genetic differences? And is the environmental variation going to be so different that we don't have any heritability? So then we cannot identify a QTL. And that may end up being an impact, like certain environments, maybe we won't see heritability, but so far we have been able to in our 2 Montana locations. And there's a whole slew of technical difficulties when we can sample the rhizosphere. If we got a really dry environment, it's really hard to get at those roots later on in development, just simple things like that. And then the way that we're looking at the data is by sequencing ribosomal RNA, and there's some issues with the sampling and the type of data that we produce that has caused difficulties as far as creating the GWAS. But these are the research questions that we're interested in addressing, and at least some of them, the data that Jessica is about to present will give you an idea of where we think we're going with this project. So I'm going to turn it over to Jessica Williams at this point and allow her to present the data. I think you might just leave it, see if it works. Let's just see.
Can everybody hear me? Yeah, okay. Great, so I'll just go over the research questions that we're trying to get at with this project to start off. Are variations in barley rhizospheric bacterial communities heritable? So can any of that variation be attributed to plant genotype or variety? Can specific barley genetic loci be associated with bacterial taxa abundance? Are any of these loci consistent across environments, which would suggest they could maybe be broadly deployed in breeding? Is there co-segregation of microbial associated loci and barley agronomic performance, which would be helpful? And do variations in microbial communities explain location-specific adaptation? So this kind of gets at the home field advantage concept that we've been investigating with that Barley microbiome research group. which I'm not going to talk about much today, but we recently had a paper come out about that in crop science, if you want to check it out. So to get at those questions, we conducted 2 field trials that I'll talk about today in Montana, in Moccasin and in Bozeman. We did these in 2021, so both were extra dry that year. Moccasin typically tends to be drier, and that year only got about 9 inches of precipitation for the whole crop year, so starting the previous fall. And in addition to these climatic differences, the 2 farms are managed differently. So in Moccasin, we tend to fertilize a little more heavily in Bozeman, and then we also use tillage in Bozeman. The farm in Moccasin has been no-till for many years, and also in Moccasin they plant filler plants in their alleys and between plots and fields. So there's living roots throughout the soil on the farm for the whole season, which we think might have an impact on the microbiome. And we saw these management differences reflected in the soil tests that we collected on the fields that we used, where soil nitrogen levels and pH, the nitrogen was higher in Bozeman and the pH was a little acidic, while we had slightly basic soils in Moccasin and higher levels of soil organic matter. We used the S2 Met population for this project. So this is a group of lines from spring 2-row malt lines from lots of different breeding programs put together by Kevin Smith and colleagues to investigate some genomic selection questions. But it worked well for our purposes because we've got lines that are adapted to different environments, and conveniently the population has already been genotyped, so that's nice. Because it's a large mapping population, we used an augmented design for our trials where we had replicated checks, but our 280 experimental lines only appeared once in each location. And we collected agronomic data on these field trials, as well as rhizosphere samples. So to do this, we dug up the top 6 inches of root systems from a few plants in each plot. And did this at about late elongation, early booting phase. We knocked off the loose soil from these root systems, but tried to keep the soil that was adhered to the roots, brought these samples back to the lab and took them through a series of washes and sonication steps, and then centrifuged everything down to get a pellet that consisted of the soil that had been on and immediately around the roots and contained any microorganisms that were in there. We performed DNA extractions on those pellets and then sent those DNA samples to a facility to undergo 16S sequencing using the Illumina MiSeq platform. And that sequencing basically just allows us to identify which taxa of bacteria are present in these metagenomic samples. But an important thing to note about 16S sequencing is these data sets are compositional. So they tell us the abundance of these different taxa relative to each other within each sample, but they can't indicate absolute abundance of these taxa in the rhizosphere. I really like this figure from Glure et al. To explain that, where we're unable to distinguish if the top graph is representing what the reality is in the rhizosphere, the bottom graph would be indicating what we get from our 16S results. We're unable to distinguish between samples 2 and 3 for the abundances of taxa A and B because they're in the same proportion to each other within a sample, even though their absolute abundances are different in the rhizosphere. So just something to note when we're talking about these types of results. So we used the relative abundance of different bacterial phyla and genera as extended phenotypes in our heritability analysis and genome-wide association study. And I say extended phenotypes because, of course, the relative abundance of a bacterial genus is not a plant phenotype. But if we treat it like one in a GWAS, then the results we're hoping could help direct us to plant genetics and phenotypes that are involved in these plant microbe interactions we want to study. Another thing, the 16S sequencing indicated the presence of thousands of taxa at each location, but many or most of these taxa only appeared in a few of the samples, which is not really useful for a genetic study. So to start off, we've applied basically a prevalence filter to our taxa lists, where we only looked at bacteria that appeared in at least 95% of our rhizosphere samples. When we did that, we noticed something interesting. A lot of the taxa were prevalent in both Bozeman and moccasin, but about 60% of the total number of genera and 33% of the phyla were prevalent in moccasin, but not Bozeman. So there are a lot more taxa that appeared in more samples in the moccasin location. Kind of going along those lines, when we look at just the number of associations from our GWAS, so looking at the graph on the right there, we had far more associations and results from our GWAS study when looking at the moccasin data set than the Bozeman data set. We kind of have a working hypothesis going for why we're seeing that pattern is because the moccasin location was the more stressed environment in terms of water and nutrient availability. Maybe the plants were under more pressure to form these beneficial relationships with the bacteria to acquire resources and tolerate stress. Also, maybe the management conditions at the moccasin farm over the years have been more conducive to building up a more diverse and overall abundant resident soil bacterial community. So maybe the plants had more opportunities to form these relationships there as well. We did find heritability of our bacterial extended phenotypes in both locations. Depending on which taxa, these ranged from non-existent to kind of conspicuously high, but the heritability would indicate that maybe this is a possible thing to breed for. So let's look at some Manhattan plots. If anyone's not familiar with Manhattan plots, each dot corresponds with a SNP genetic marker. The x-axis indicates that marker's position in the Barley genome, and the y-axis is telling us the strength of association between the phenotype being graphed and that marker. When we looked particularly at the moccasin location, we were encouraged to see these patterns where different bacterial taxa were associating with the same set of genetic loci, and that gives us more confidence that these results might be real and also indicates that these loci would be potentially important ones to investigate further to try to figure out what's causing these connections. We specifically wanted to look for patterns that were consistent across both of our locations, and we did find some examples of that. So these graphs are representing 4 different beneficial genera involved with the functions Jamie was mentioning earlier, like nitrogen fixation, phosphorus solubilization, pathogen protection. And the top 2 graphs come from our Bozeman location. The bottom 2 come from moccasin, and you can see there's some overlap with which genetic loci these genera are associating with. And that's encouraging because that's the sort of thing we'd like to find to use in breeding, so that ideally the plants would be able to utilize beneficial microbes that are in the soil, regardless of which taxa happen to be present at whatever location they happen to be planted in. We also specifically looked to see if we could find patterns of co-segregation between our bacterial phenotypes and agronomic traits, and found a couple examples of that in each location. Here we have the genus Arthrobacter on top, which It's a plant growth promoter and pathogen protector in other research. And the effect of the minor allele at each of the loci indicated here was in the same direction for the relative abundance of Arthropacter and for grain protein and plump grain. So depending on which locus we're looking at, as the proportion of Arthropacter present in the rhizosphere community increased, so too did grain protein or plumpness. So we might be able to utilize Arthropacter or utilize these loci to enhance our grain quality, hopefully. We also saw a pattern like that in Bozeman, but here I've got phyla in the top two graphs, whereas the other bacterial taxa we've looked at have been genera. So with these, it's a higher taxonomic level. So there are lots of subtaxa within these phyla that could be doing all sorts of different things. Mixococcota on top there is one of the microbial predators that Jamie was mentioning. So bacteria preying on other bacteria. Maybe that plays a keystone role in the community, cultivating the presence of kind of a community structure that's important rather than specific taxa being important. Or maybe they're preying on pathogens, which would in turn be helpful to the plant. So it's not always super straightforward to think why these bacterial taxa could be helping plant growth. And it's good to remember that the whole situation is quite complex. We've got these microbial communities and the plant genetics and the abiotic environment all interacting to produce these connections that we're going to need to learn more about. But we did find heritability. We did find relationships to specific genetic loci. So it could be possible to perform selection on these and incorporate this into breeding, but we have more research to do. We have additional location years that we have yet to dig into. We also have fungal data that I'm excited to start looking at more. And recently got our sequencing results for those bulk soil samples that we were talking about that will help us to understand more how the role that the plant plays in cultivating this novel microbial community around its roots from the source that is the bulk soil. And as we identify the genetic loci that we're most confident and interested in, we're going to zero in on those areas and to search for gene candidates to try to start understanding the mechanisms behind these relationships so that we can leverage these loci in breeding to hopefully produce varieties that are able to utilize this naturally occurring resource to have more resilient crop production in sustainably managed systems. And with that, I'd like to thank you all for your attention, and thanks to Amba for letting me speak, and we'll take any questions.
So with that, are there any questions for Jessica or Jamie? Ryan in the back.
Look at the fungal to bacterial ratio in those soils at all?
Repeat the question too.
The question was, have we looked at the fungal to bacterial ratio? We have not, and I'm not sure that's something we'll really be able to do with the type of results we have, since we're not quantifying amounts of the different microbes present. But it's certainly something that would be interesting to do. And maybe we could look at ratios of I don't know if we could look at ratios of the relative abundance of particular taxa in both the bacterial and fungal data sets. Yeah, I feel like the possibilities are endless with the results we're getting.
It might be something you want to look into, because I know as your fungal ratio increases, efficiencies increase with carbon cycling, biomass creation of the plant. Might have something looking.
Yeah.
Great. Yes, Lance.
So, when you talk about the tribe, what does it look like in the present or absence of the bacteria? Or is that the present of a specific bacterial species? Or is it some boundary bacteria present in
Yeah, so the question was with the bacterial phenotypes we're looking at, is that like a presence absence of the bacterial taxa essentially? It sort of is. We know a little bit more than that. It's not a totally quantitative method. We kind of know a percentage of that, you know, like of all the bacteria in the sample, what percentage is that taxa? But it's not exact. There are other things complicating that, like different bacterial taxa have different copy numbers of the 16S gene. So it is a little fuzzy, but it's a little more quantitative than just presence absence.
One more question?
Yeah, sorry, just a follow up. For example, the constitution of biology CD appears to know those genera identified as human decisions. What's the level of the abundance?
The level of, sorry?
The abundance. Positional abundance.
Oh.
Thereupon. Okay, let's bring it.
So I think you're asking for at the specific SNP associations we were looking at in the graphs, what bacterial abundance is associated with those points. It depends on which point we're looking at. That gets into the, you know, I kind of didn't talk about it much, but I had the plus and minuses on some of those graphs about the effect of the minor allele. Sometimes it's indicating that there's more of that taxa present. Sometimes it's indicating that there's less. It's just, we have to kind of go behind the scenes and see which of those things it's mapping.
So what we know is that both alleles are present in the population. There's an allele that would increase the relative abundance, and then there's the opposite allele that would decrease it. So for each one of those hits.
Great, well, thank you. Thanks. All right. Last presentation before lunch, and I'm gonna just undo this here.